Welcome everybody to the next session which is named Precision Medicine and Biobanking. I guess we all know the rules now for the speakers. Let's try to stick to the time, 15 minutes for each speaker, and we decided that we will have the questions after each talk. I think there is plenty of time also devoted to the question and answers. It is my pleasure to introduce the first speaker, Lars Baumbusch, who is a senior scientist at the Department of Pediatric Research, Division of Pediatric and Adolescent Medicine, Oslo University Hospital. I know this is not the first time you are in Riga, so welcome.
Yeah, thank you very much. It's actually a very great pleasure to be back here in Riga. So we have heard a lot about pediatric cancers in the previous session, so I won't repeat most of it. So in Norway, we have about 200 cases every year, making it to the dominant cause of death by disease among children above 1 year of age. And despite that there are good treatment opportunities, we still have very different high-risk subgroups with relapse or resisting disease. And we all know that childhood cancers are different from adult cancers. They have different cure rates, they have different genetic background and different frequencies. So what you can see here with the adults, we have the breast and prostate cancer and lung cancer, but the childhood cancer, we have ALL and brain cancer. So we started our initiative actually when we read about the American Pediatric Cancer Genome Project, which was established in 2010. So about that time, we became aware at our university hospital that for the adult cancer, there were different biobanks, different sequencing projects available and up and running, but there was nothing like that for childhood cancers. So there was a lack both of the infrastructure, but as well, especially a general informed consent. So at that time, when the parent came there with their child, they had to sign different papers for different projects. And if you are in this vulnerable situation, it's of course crucial if people coming with different papers. So we said, okay, we need one general informed consent, and that will then lead to all the different research projects. So we started then with our biobank, and I think that's also very crucial. We had some people which were dedicated to it. So I would say the requirements for a general biobank is that you really have people who see the demand, who see what is necessary, and that you also have an administration supporting it. The next step is then you need collaboration. So of course you have different departments and all of them have to work together. You have the oncology ward, you have the pathology, but you also need lab facilities who can treat and handle the different samples in the correct way. Before you start biobanking, you need approvals. You need the ethical committee to approve for the biobanking, you need the administration, you need a data safety board, and you need operation procedures. But you need also an infrastructure. So you need people who know what is biobanking, how to treatment. For example, we are treating all of our samples within 1 hour. So when you get the samples, everything is then aliquoted and put into the freezer within this time frame. But you have to be aware of these kind of things if you are working with biobanking. You have to be aware how to register a patient, how to handle things, and how to store it correctly. And of course, you need funding. And that was also a problem for us in the beginning, that we needed some funding who support the biobanking asset. So what we built then is a consortium. And we started actually first at our university hospital in Oslo. So we started here and we got our infrastructure up and running at our hospital. And then after some time, we invited as well the other university hospitals in Bergen, in Trondheim, and in Tromsø, and all of them contribute now to the biobank. And what was also very important was who owns the biobank. So now it's a share between all the university hospitals, or it's in a way in consortium itself. So it's not that the samples do not belong to Oslo or to one of the other hospitals, but it's a combined effort and a combined utilization of these samples. And we have an advisory board which looks at the scientific questions, and we have a steering committee who meets twice a year. And what you can see here, so we are collecting at all of the different locations, and some of the special treatments are only done in Oslo, while the others are storing at their locations, and then everything is transported to Oslo for further work on the different downstream analysis part. So if you look at our pipeline, of course, in the beginning, you have the informed consent. And that's also a question often. So in Norway, it's compulsory that both parents are signing the informed consent. And that was during the pandemic, it was quite difficult then to get both parents at the ward. Then you have the registration and the transport. And then is the next question, what to collect. So we try to collect really everything we can get hold of. So from blood, urine, stool sample, and hair, whatever we can get hold on. So that's now our pipeline. So we have the different university hospitals. We have a registration at each site. Then we have the processing and in the control lab. And then we have the long-term storage. And then we have the different research trials and translational research. So then we have the exchange, again, with the hospital, depending on which research question they are working on. This is to show you how our registration looks like. So that's on a separate server. And that's the different topics which you register, so gender, ethnicity, metastasis, and other medications, and so on. So we have quite a long list what we are registering in our database. This is our status today. So we have now included over 730 patients. And we are very proud of that because over 95% of all patients which are asked to contribute are willing to do so. And of course, the majority is ALL. We have AML, and a large portion of solid patients. And it's a little bit hard to read, so we have in one of our first publications, which came out last year, we have also looked at the 500 first patients, so where you can see the distribution of the different solid tumors seen as leukemia and other types. That's what we are collecting. As I said, we are trying to collect everything. Full blood, plasma, serum, Buffy code, and that makes it also different from other biobanks, for example, the adult biobanks. Some of them are only collecting the tumor material and blood. We have also in addition bone marrow when we can get hold of it, spinal fluid, tumor tissue, salvia, urine, feces, hair, germline DNA, tumor DNA and tumor RNA. And that was also a question in the beginning. Should we extract it up front or should we first collect it and do the extraction later on? And we have a new project where we're now collecting living cells from the bone marrow and to put them in the -150 freezer to have living cells from these patients where we can get hold of. And today we have 70,500 samples, which we can now use for research. And I mean, one thing is that we have these samples, but we are also very proud that we are one of the first national biobanks. So most of the adult cancer biobanks are only localized in one hospital, but we are really nationwide. And that took some time to get the trust that we can really collect everything together at one place. That's our homepage, so where you can get access to it, and then you can see we also have the different projects located there. And these are one of the first projects which we started. And I want to introduce to one of the projects, which is called Inform. And so there, Monica Montegos is the PI of it, and The biobank, that we have the infrastructure of the biobank, was a real prerequisite that we could do or be part of this project. So in form, we will hear more about it, I think, tomorrow, is an extended genomic and molecular characterization of children with relapsed or refractory cancers. So where you have come to an end of your therapy, and the hope of it is that you get improved treatment through participation in targeted clinical trials. So what we are doing, we're using the infrastructure of the biobank, we're collecting the samples, and then it's sent to Heidelberg, where the rest of the analysis is done. But you can only do that if you have the infrastructure of the biobank. What I would like to discuss with you today is also how to continue. Because now I think we are in a kind of a crossroad where we're discussing internally as well how to continue, and that I would like to share with you. For example, with the DNA and RNA extraction. So we are doing that now routinely, but we all know that if you put the RNA sample in the freezer, it's not as stable as DNA, so is it really wise to do that? What about sequencing? At the moment, we can't afford upfront sequencing, but it would be wise to do so. Should we then start with exome sequencing? Should we start with whole genome sequencing? We don't know. Quality control is a big issue for us. So we have now started with some quality control for DNA. So there we have an experiment where we have put in a large bulk of DNA, and we're taking out a DNA sample every 3 months or every 6 months, and we are testing the quality of it. But how to assess the quality? Is it only the amount? Is it that you put it on a gel? Is it that you're looking for some specific mutations? It's hard to know. And then as well, should we only look for DNA? Should we be starting with the same for RNA? Maybe we should do it for protein or metabolomics. So how to know that in 5 years from now on, or 10 years, your sample which you collected today is as good as it was when you started with your biobank? Long-time storage. You see here this fantastic facility. At the moment, we are only storing it in one facility, but we also got the offer to share a place in a robot. Should we do so? What would be the best then to access the samples in the future? It's really hard to know. Should we be part of a larger biobank, like the adult biobanks, or should we try to keep it separately? Childhood cancer is different. We're collecting it differently. We get smaller amounts at different time points. So this is quite challenging for us to get it through. Financing. At the moment, each new project has to pay around 1,000 euros to start it for the biobank. In a way, it's nothing if you look how much we spend for the freezers and for the staff and so on. Should we have a different system? How to put fees on the people which are using our samples? How to make that fair? Should it be cheaper for national-wide use than for international use? I don't know. And then as well, how to guarantee that we can use our samples in a wise way, in a good way? How to share it when we get requests from international organizations? How can we put up good organization? How is it with the data return? If one researcher is running a sequencing project, is he obliged then to send the data back, and in which time frame? Or should we do it for him or her? And the other thing is, how should we use the limited material? Often you have only a needle biopsy of a patient. Should we wait? Should we collect it until we have a certain amount of patients? Or should we use it now, today? And that's maybe a typical Norwegian question, because as you know, we have a lot of money from oil and gas, but we're putting into the bank, not using it for investments. Maybe that's the wrong way. Maybe we should start to use not only the oil and gas, but also the biobank material today. I want to summarize to say biobanking is more than just collection of some samples. What we learned with our biobank is that the synergy effect which we had with our colleagues, that we are really trying to have a combined research effort of the different university hospitals. And that's what is so brilliant, or what is really encouraging us to continue with this work. But as I said, we are now working on how to use the samples in a good way, how to use the infrastructure which we have established so far in a good way, and to keep on with the synergy effect of our collaborations. And we are asking ourselves what should be the focus, the aim, and the strategy for our bank in the future. And I want to end with to thank my colleagues. So Monica is a PI, and I'm in the co-PI of this biobank. Then we have a study nurse and coordinator of the biobank, Nina, and we have a technician, and all of them are very dedicated to our biobank. And here you can see all the different people from Trondheim, Bergen, Tromsø, and all working together. Okay.
Okay, thank you, Lars. Okay, now the floor is open for questions. Okay, maybe I can start. I know how difficult it is sometimes, especially with initiatives where you don't get a lot of funding up front. It's difficult to convince all the participants to really collaborate. Was it easy? How it was in your case?
Yeah, it was difficult, and it is still difficult. So at the moment we are financed mainly through the Childhood Cancer Society, and they financed our administrator and the study nurse for 3 years, but after that we don't know. And we are discussing it with our clinic, because we think that a biobank should be part of a clinic or should be part of our university hospital. But of course we have, I think, 20 to 40 different biobanks. It's difficult to finance all of them, but we are negotiating that we get more funding and more staff for the biobanks. But then again, if you get financing support from your clinic, you also have to show that you are using it, that you are producing publications, that you are coming out with research success of your biobank. So I think it's also, yeah, you are obliged then to show that you are using the money wisely.
Okay, yeah. Oh, there's a question.
Yeah, thanks, thanks, Lars. Wonderful presentation. I will ask again provocative question based on the slide which you show perspectives for discussion, and you posed really important questions. Now my question is, how are you going to find the answers for those questions, and how long it will take? Because the question is, are you going to do that in silo on a country level to find those answers, or are you seeking some international perspectives and consensus?
Yeah, so actually I'm just coming back from a meeting in Tromsø where I meet some of my colleagues. So we have this steering board committee, and I think inside the steering board committee we have these discussions, and there we decide in a way, and there we have representatives from all university hospitals. So we decide in a way together what we want to do and what we want to achieve for the future. But of course, if you have 10 different people, you have several different opinions. So some want to keep it in the biobank for the moment, and others say, okay, let's do some more research, even if we are using up some of the samples. So both sides. So I think we come to an agreement in the end, and maybe the majority then will decide what to do.
Okay, yeah. Next question.
Yeah, thank you for the very informative presentation. So I was wondering, as INFORM, for example, is a study, of course, for the relapsed refractory patients. So how are you now linked with this adult precision medicine studies and clinical trials, which we will hear more about tomorrow in Norway, and what is your future plan for that, also for the primary cancers? And yeah, would be nice to hear.
So actually, so the Oslo University Hospital, it's in a way there are 3 different hospitals. So one is a radium hospital where they only treat adult cancers, and we have the Rikshospitalet where we only treat childhood cancer. So in a way we have 2 different localizations, but we try to interact, of course, with them. So we are part of some of studies there, so we have interaction, and what we want to do in the future, I mean, now we are part of Inform. Inform is brilliant, but we have to bring it back to Norway. We have to learn our lesson, and we have to establish, in a way, a pipeline which is similar to Inform. I think it's important to have that as well in-house. So we are trying now, we are running it a little bit in parallel. So some of our samples are done both in Heidelberg and in Oslo, and we're comparing the results, and we will see if maybe we manage in the future that we get it as well established there. But of course, 200 samples, it's not that many, or 200 cases is not that many. We still need the expertise from others.
A kind of additional question, promoting one. So as we have now here many people from Nordic and Baltic countries, would you see that there would be a kind of place for collaboration as well?
Of course, absolutely, absolutely, yeah. So I forgot to mention, I mean, the next speaker is from the Swedish Childhood Tumor Biobank. When we started, we went to Sweden. We asked them, how are you doing it? We learned a lot. So we are very, very thankful to the Swedish Biobank. So they told us that they do things a little bit differently, but without them, we couldn't have started.
Okay, thank you.
Yeah, and with this word, I can introduce the next speaker. Yeah, which is Johanna Sandgren. So I'm very happy to hear you, because I know that you're doing things a little bit differently, and I think that's important, that we're not doing everything the same way.
Yes.
So please.
Yes, thank you very much, and that was a very good transition also to my talk. Thanks. So I'm going to talk about the Swedish Childhood Tumor Biobank, and it's called
I think we can't hear you.
Is it on?
Yeah.
Stand closer, maybe. Oh, sorry. So it's called Barntumorbanken in Swedish, or we usually say BTB. So it's a national sample collection and a genomic characterization initiative of pediatric solid tumors for research purpose. And I also have an introductory slide about pediatric cancer. So in Sweden, we have a bit larger population than Norway, so we have 340 children every year diagnosed with cancer. 1/3 are leukemias and 2/3 are different solid tumors. And today more than 85% of these patients survive, and it's still though the major cause of disease-related death among kids. And I also have some survival curves here for several decades back, and you can see for several diagnoses has been plateau here in regards of survival. And also the survivors often suffer from side effects from the current treatment. And moreover, as you all know, I guess, childhood tumors differ from the adult forms. They have different biological origin because they occur during development, and they also in general have much less genetic aberrations and also seem to interact less with the immune response. So overall then, of course, more research are needed on these malignancies. So that's the big reason then to form the Swedish Childhood Tumor Biobank, and that was around 10 years ago. So as Lars said, we started some years before in Sweden. And here are some points then about BTB, the Tumor Biobank. So it's a research and healthcare-oriented biobank infrastructure. It's situated in Stockholm, Karolinska Institutet. And it's a natural sample collection for whole Sweden for pediatric solid tumors, and we collect most materials as fresh frozen. And the sample collection was established at the Karolinska University Hospital in Stockholm. And what we do is also then genomic characterization of the collected biobank samples. And one important aim then for the biobank is to be able to distribute these samples and also the generated data to research projects. And we are fully funded by the Swedish Childhood Cancer Fund and has been all over the years. So the overall aim is then to support research to increase the understanding of these childhood tumors so it can lead to enhanced survival and quality of life for these children. So some more details about the BTB. So of course we are a biobank, so we register the samples. We have a LIMS system for that. We prepare and store them. And we do then the molecular and genomic analysis. So what we do is the whole genome sequencing of the tumor and blood samples. We do RNA sequencing and also methylation arrays. And then we also develop an informatic pipeline so we can interpret the results from this data. So we have a pipeline for whole genome sequencing called SARIC and different pipelines for the RNA data. And that we do together with this big life science infrastructure in Sweden called Science for Life Laboratory, which also do the sequencing for us. And what we provide is then service to different clinical study, collaboration to researchers, but of course also we have this formal process, so researcher can apply to us and get access to the samples and to the data. And of course you need an ethical permit for your research project. And then we then have this crucial collaboration with the 6 Swedish university hospitals that operate and treat these children in Sweden. And you can see where they are on this map of Sweden. It's different cities here, and then we collaborate with the different departments, the pediatric oncology, the surgery, and the pathology to receive the blood samples, the informed consent, of course, and the tumor tissue, and also some clinical information about diagnosis. I will tell you some more about the sample types later. So what about the sample collection? So as I said, we had been ongoing for a bit more than 10 years. So here you can see some bar graphs of how many cases we collect each year, how many patients. So we started with CNS tumors, the blue part of the bars, and then we continue with other solid tumors around 2015. And we collect from almost all children with a solid tumor in Sweden. We get the biobank sample and a consent. And here is also a table for the status about 1 month back, that we have overall collected 1,860 cases. And we also have written here that we have paired blood and tumor cases from 1,173, which is important when you do whole genome sequencing. You need a germline and a tumor sample for that. And we also have the numbers for the different centra, and of course they differ depending on how many patients are referred to the different centra. Some more words about the sample collection. So that was the number of cases. And we have now around 23,000 samples collected, since we get several samples from each patient and also different sample types. And we do DNA and RNA extraction in the BTB lab. And so we have, in addition to those samples, then almost 21,000 aliquots of DNA and RNA in the freezers. And some more words about the sample types in the other part of this slide. So as I said, we collect fresh frozen solid tumors, which is important for the DNA RNA quality since we're doing the genomic analysis. And we collect the patient-matched blood samples. We also collect CSF. We also accept some formalin paraffin-embedded tissue, collect some diagnostic slides, and we also get fresh tumor from some centra so we can isolate viable tumor cells. And we also can collect some hair and saliva as well. And also we collect the blood from all the parents, which is important if you're interested in the inheritance of the cancer disease. And lastly, on this slide is also that we have ethical permit and patient consent to collect CNS reference cases, which are then from patients that are not suspected to have a tumor, a brain tumor, and they are operated due to other causes like epileptic seizures. So this could be like a reference tissue for brain tumors that could be age match also. So we are then a population-based sample collection of solid tumors in children. So the pie charts here show the proportion of the different diagnoses in the biobank for the CNS tumors and other solid tumors that fits well then with the incidence of these diagnoses. We'll now switch a bit to the genomic part of the work. So we'll be mostly talking about the biobanking and sample handling. And as I said, we do the different genomic characterization, the whole genome sequencing, RNA sequencing, methylation arrays. And then we collect a lot of big data that we need to store and process, of course, and we do that mostly internally at KI, but we also use some external infrastructures in Sweden for this. And then we do the bioinformatic analysis, as I told you, and then we have data sets that we can and have now also distributed to authorized researchers when they apply to us. But I also want to mention here that we, since a few years back, also collaborate with the Genomic Medicine Sweden initiative that is then to implement the whole genome sequence in the clinic that David Gisesson talked about earlier today. So BTB is also part of this study, and then we work closer to the hospital. And one important task for us is then to also to store this generated data for secondary use in research. So that data also is deposited at BTB. So how much data do we have now? So this is only the BTB data, not the GMS data. So we have sequenced around 600 cases, and this is ongoing. We will do another 150 this year. So in these bars, you see the whole genome for the tumor and blood and the RNA sequencing. And around 400 cases, we have profiled with methylation arrays, since we only do that on CNS tumors, and now also sarcoma, since they are also a classifier for those diagnoses. And I also mentioned the INFORM study here, as also Lars talked about, since 2016 in Sweden are a national hub for those patients in regards of handling the samples and sending them to Heidelberg for this molecular genetic analysis. And the idea is also here that we also can actually get this data back, because they are doing similar analysis as we do in the biobank, and then we don't have to do the double analysis with the piece that we have in the biobank, so we can save material and share data. But there are some legal issues to this also, to share data, but that is the plan, and we have done more than 200 of those patients for the INFORM study. And here I also mentioned the bioinformatic pipelines that we have that are also open source. And also one slide that we are preparing a manuscript about the Swedish Childhood Tumour Biobank to describe the work and the workflow. And also we have done an in-depth analysis of 82 CNS tumors to show the research and clinical utility of this. So this is one figure from this manuscript showing all the 82 different tumor cases and the important mutations found in them. And I also want to mention then, since I said at the beginning, important then to distribute the samples and the data to a researcher, and we started to do that some years back. So we have this process for this, we can apply to BTB, and there will be a scientific evaluation of the projects, a medical legal review, and of course we check if the sample data are available for this project. And so far we have had 10 approved research projects within Sweden. So this is a table of the 10 projects going to different universities in Sweden with different applications and different projects. There are methylation studies, there are spatial gene expression, single-cell RNA, xenograft studies, cell-free tumor DNA studies. And they have received tumors and data from BTB. And we have more applications ongoing, and this is something we will work more on now to receive more applications. So the data and samples can actually be used for research progress. And we also then assist different clinical studies in regards of sample handling and also some genetic analysis. Like the INFORM I mentioned, we also help a cerebellar mutism syndrome study, where the study center is in Copenhagen. And as I mentioned, this GMS childhood cancer study, which is a natural study. We will also aid 4 more studies this year. And these are all international or natural studies that we support in the field of pediatric cancer. And previously we have supported the Biomedical and CUPNET studies. So, and of course we are several dedicated people working with the biobank. They are listed here, and we have the good collaboration with all the 6 centers that we also have on this slide. And of course, we're also dependent on external collaborators and the funding agency, the Childhood Cancer Fund, over here. So, thank you very much for your attention.
Okay. This paper is now open for discussions. there any questions from the audience? If not, I want to start with one question. So where do you see your future for the biobank? So will you continue sequencing all of the samples you get, or do you think you will take a selection that you only take relapse samples or so on? So where you see yourself for the biobank in 10 years?
In 10
or
years?
5 to 10 years, yeah.
That's a good question, of course. So now the focus is to sequence what we have. We also got this special grant from the Childhood Cancer Fund to sequence what we have in the freezer. But now, since also the sequencing is done in the clinic with this GMS study, we of course will not do it on the same sample in the biobank. So we're now discussing which other possibly methods we should use on the biobank samples, what the research community need. So of course, that's something we continuously think about, and also collecting different sample types, and yeah. So it's a good discussion, but we haven't decided yet.
Yes.
Hi, thank you for this great presentation, and I got particularly curious about that fresh frozen material part. How do you manage the logistics to get the material immediately frozen and then transported, and how does this work?
Yes, from the 6 different centers also. Yes, but we receive them in batches because this is for research, so it's not for clinical use. So they freeze the samples locally and then them send us 3 or 4 times a year. So it works fine, actually.
And why? What is better than taking a material in like a stabilizing fluid? Like tumor material can be also stored for DNA extraction in just like a tube. And what is better in frozen?
Right now, they store it in a tube, so we get the frozen tissue to us. But we want also to be able to freeze it in media, so we can also have viable cells in the tubes, but that requires some more protocol than at the local centra. So right now they just freeze the tumor piece in -80 and send those to us on dry ice.
Thank you.
Thank you.
Are there any more questions? If not, I would like to announce the next speaker. So thank you very much for a wonderful presentation. Yeah, the next speaker is Jānis Kloviņš, here the chairman of the Scientific Council of the Latvian Biomedical Research and Study Center, and as well as senior researcher of the Latvian Biomedical Research and Study Center, and a professor of molecular biology here at the university. The last time I was here in Riga, I was shown around at the biobank, and one thing which impressed me most was the safe. So there was, I remember there was shown that it was previously a bank or something, so you had this huge safe for the samples, so that really impressed me. So, please.
Okay, we now move to another building, so it's a bit different now, but yeah. Okay, after having heard those really nice presentations from the morning session and also today, I'm very happy to present the Latvian case. So, and I will talk about the Latvian Pediatric Cancer Initiative, show some pilot project results. First of all, I would like to mention that I'm speaking on behalf of Monte Brīvība, who is in charge of this project. She is a coordinator of this project, but since she is now at home with a 2-week-old baby, so I'm presenting this talk right now. The project I'm talking about was devoted to really investigate what are our technical capabilities and how we can actually work towards to bring this precision medicine strategy in the treatment of pediatric malignancies. More specifically, the main goal was to develop the workflow similar to what we have heard today from many speeches for molecular characterization of pediatric malignancies. And of course, the objectives are straightforward. The one objective was to establish the cohort of pediatric oncology patients, another perform large-scale genome analysis, and then try to establish a working group to really test and look at the possibility how these data can be used and whether they can be used in clinical practice. And of course, when you look at the tasks, they are very similar to other similar projects. So basically, you have to perform recruitment of the patient, starting from the collection of clinical material. Of course, also all the legal documentation has to be there, sample processing, sequencing, finally data analysis and interpretation. And now looking back on the results, actually, I'm quite amazed how fast we were able to organize this project, because I first met with John Tully when we were discussing to really how we can fund this project in February 2019, and already in the October 2019, just a month after we have received ethics approval, we were able to really show the first whole genome sequencing report, and then the things were really moving quite fast. But the reason why it was so actually was because there was a really collaboration between different institutions. First of all, I should mention the Children's Clinical University Hospital, providing all the recruitment of patients and collection of clinical material. Biomedical Research and Study Center, which I'm representing, was responsible for the biobanking, sequencing and data analysis, together with also scientists from many universities in Latvia. And last but not least, we also managed to have on board some private companies like Smart Omica that actually provided some data analysis and interpretation for some of the samples. And interesting project that was developed during the course of this activity was establishment of the Genome Data Network, where MikroTik and mobile telecommunication provider LMT was working together to establish this Genome Data Network that I'll be talking in a minute later. But of course, you need also infrastructures to do these projects. And the reason why we were so fast and efficient was that already from 2006 we were working in frame of genome database of Latvian population, which is National Biobank, starting from 2006, which is now already included more than 38,000 participants in it. And the reason why we were able to do it so efficiently also was that really all the procedures, starting from the sample collection, ethics permit, and ethical approval regulation was already in place from the very beginning. So then basically all the technologies were there also for the sequencing and other processes. I'm showing you just a few pictures from our new facility that were recently upgraded the sample storage system here, the biobank, also the sequencing facility or sequencing laboratory. So you see the two main sequencers. This was the sequencer that was used for this project, and now we have also installed MGI T7 instrument that is in output more similar to Illumina's NovaSeq, which will definitely also make this process cheaper and possible also faster. Another important thing that is also having the private partners on board, I would like to specifically mention the MGI the company that provides the high throughput sequencing technologies also for this project. And I already mentioned the Genome Data Network that was initiative from two companies, MikroTik and Latvian Mobile Telephone, that really created the fast data network between the major academic institutions that were connected with the sequencing facility, and also the high performance computing center, allowing to enable these data transfers. Another important initiative is that these local activities are also well linked together with the European initiatives, and here I'm showing the recently started project from to establish the Latvian genome variation reference in frame of the Genome of Europe that is now funded from the Recovery and Resilience Facility, where we are aiming to sequence more than 3,000 whole genome sequences from general population that can be used also in all projects since the population-specific reference is really needed for many genomic projects. More on the project results. So at the beginning, we established the workflows and procedures for different tumor types, and more specifically, so for the liquid tumors, we are collecting from the blood, we are directly isolating DNA, also having the plasma and serum stored as a reference. So DNA for the liquid tumors, we are using saliva sample and the DNA isolated from saliva sample. And then depending on the cancer type, we have either lymph node biopsies or the bone marrow biopsies or material that we use both for the DNA and RNA isolation that is then processed through the to the next generation sequencing, data processing and interpretation. That's for the solid tumors, we use the blood as this reference DNA for the genomic DNA, and comparing the DNA and RNA isolated from the solid tumor, then the workflow is the same. And all these samples then are stored in number of aliquots in our biobank according to the procedures that we have established. So what are the actual project results? You already saw in one of the previous presentations that we managed to involve close to 200 patients. So we have at least 181 patients from the oncology department at Children's Hospital involved in this project. And knowing that this project was started in 2019 and we have around 50 patients, new patients every year. So you see that we are really have included the absolute majority of childhood cancer patients in our project. So the NGS, the whole genome sequencing analysis, has been performed for 85 patients. And for the cancer samples, for the cancer tissue, we do at least 70x coverage using 150 base paired end sequencing. For the DNA extracted from the normal tissue or blood or saliva, we use at least 35x coverage, while for the RNA-seq we do at least 100 million paired end sequencing reads to analyze them further. And for the specific needs of this project, we decided to use commercial software to really to get these results in more standardized manner. And in this case, this is a QIAGEN CLC genomics workbench. For some more specific variants like structural variants, we have the pipeline developed in-house, and similarly for the analysis of fusion genes, we also use the same tool mentioned before. So an important thing, the next thing is of course to bring these results to the clinicians, and we were initiating this discussion of findings basically as a training to really see how these data can be applied to the clinical needs and to really have this connection between the molecular biologists and the clinicians from the other side. So discussing the clinical implications, looking at the viable clinical trials, both using the QIAGEN clinical insight reports, and also in some cases the Smartomic provided in-depth analysis for some more for number of samples to go more deeply into the results. So of course the sequencing data are also viable for research studies, and here you can see some statistical data from the sequencing that we have done so far. For example, we have identified more than 192 pathogenic mutations that can not always were included in these reports that are provided with the commercial tool. So this is additional information that can be used either for the research studies or also to complement these clinical reports. Another study, as an example, we are now studying the fusion genes in all hematological cancers, where you can see that there are different amount of these fusion genes at different cancer types, and also looking at the most frequent fusions, from which some of these are really known as related to the cancer cases. So at the end, I would really like to point some achievements. So basically, we really met our goals. We established the most comprehensive national cohort of oncology patients in Latvia. We were able to ensure the whole genome transcriptome sequencing for the large proportion of patients, and also started to establish this framework to facilitate the use of data for clinical practice. As a future plans, of course, and this is one of the occasions here, to really continue to establish collaborations with different institutions or consortium from abroad to doing the same thing, and then learning and exchanging this experience. From the we also, of course, looking to improve some sample processing time, which is something that is really needed if you want to have these data to be used for the clinical purposes. And we are planning to include some other sample types like stool sample, and then perform metagenomic analysis. And of course, as already was mentioned in one of the previous talks, that the establishment of the molecular tumor board that could be one body that can use these results is an absolute necessity in Latvia. And on the last slide, I really would like to thank specifically people that were involved in the sample collection and the processing of the samples, and of course the main founder, Mikrotik, and John Tully personally, who really promoted all this idea on establishing this biobank and study. Okay, thank you.
Thank you, Jānis. Questions from the audience? On your last slide, you showed data sharing for future plans. I mean, in Norway, we have a lot of restrictions when it comes to data sharing. How is it here? Is it easier to share data with other international research groups or inside the clinic?
So far, there is a regulation in the law that in order to really send the data or samples abroad, we have to go through at least 2 committees. So basically, it's a genome analysis committee, and then of course the ethics committee. So in principle, it is possible. In the future, I really look on the initiatives like the 1 Million Genome Project and GDI network that is now actually developing these practices for the federated data storage system. And if such a thing would be in place and regulated with the common regulation from the Europe, I think that would really put all the things in place and would really facilitate the data sharing that would be more regulated.
Okay, so I just have to admit, I think it's really impressive how much you have achieved in so short time. I mean, it took us several years to get our biobank established, so I think it's wonderful.
I was a bit surprised too, but that's really thanks to these people and specifically Monta, who really was organizing all these activities. Yeah. Thank you. Okay, and then our next speaker is Eero Punka from Helsinki, Director of Helsinki Biobank. So please, floor is yours.
Thank you. So regards from Helsinki, and thank you to the organizers for this opportunity to give a talk here. So I'm going to be telling you about our ongoing effort of returning clinically relevant data from biobank projects to healthcare. So this is our hospital, Helsinki University Hospital, where our biobank belongs to. And as you know, as you see from the numbers, there's a huge potential for biobank samples and also biobank data. And if you see here almost 3 million patient visits per year, and you know that in Finland the lab values, for example, have been recorded digitally since 1980s. There is a lot of data available in data lake infrastructure, which has been made at our hospital. And I just recently heard that our data lake is the largest clinical data repository in Europe currently. Helsinki Biobank has a population base of 2.3 million people, so about the same as Latvia, I think. And that's because most of the people are living close to this southern coast. We are owned also by Helsinki University, the University of Helsinki, so there is a close collaboration between the university and the hospital. Before going to the subject, it's really important to understand the rules of the game. And in Nordic countries, the biobank law is quite similar, but there are differences. And in order to really return data back to the donors, it's good to know that the law has to be clear. The ethical sides and also the rights have to be there in place before you can really do it. And in Finland, the biobank law has been in operation for about 10 years, and it has proved to be quite progressive. So there is a wide consent that we get from sample donors. We can transfer existing collections to the biobank, and we collect samples from as a routine from healthcare during the normal procedures. We can combine this rich real-world data with the samples, and very importantly, we have the right to recontact the patients, so we don't have to hesitate. If there is a reason, we can recontact the patients, and we can also serve the industrial customers as well. So looking at the biobank model in Finland, it positions us between the hospital and research. And this is very good. So we are getting consents and samples and data from healthcare, and then we are delivering that to researchers. But I would like you to focus on the lower right corner, which is also dictated by the biobank law, that we have to, if the patient asks, we have to give the data back to him or her. And if there is some clinically relevant information, we should tell them. And this is tricky. How do we know what is clinically relevant? This is our consent form, and the red arrow actually shows the place where the sample donor should tick, so that she actually, or he, gives us the right to inform him or her when we find some clinically relevant information from the sample. And 99% of the people actually tick this box and also the following box. And although this is an informed consent, we understand that people don't really understand what they are ticking. Then I tell you a couple of slides about FinGen project, which is the largest biobank project in Finland, and it's also one of the largest genomic projects worldwide, where we are collecting 500,000 consents and blood samples and DNA samples from Finland. So this project is run by Helsinki University, and our biobank is responsible for coordinating the nationwide sample collection. All Finnish biobanks are actually participating in the sample collection. And it's a GWAS, so genome-wide association analysis study. It's also sponsored very greatly by pharma companies. There are currently 13 pharma companies participating in this research. And the genomic data is then compared with the health register data and some hospital data. The FinGen project has been running nicely. It started in 2017. We have already delivered 430,000 samples to the project. And so we are in 6 months or so, we have 500,000 delivered. So that's a very good situation for the project. And yeah, sorry, wrong slide. So we have now almost all samples collected. But the problem comes now. We have by law, we need to deliver the clinically relevant information back to the participants. And of course there is, because it's genomic data, it has a relevance. And currently we have got 300,000 patients' genotypes back to the biobanks. And now we are developing together with the hospital professionals and the geneticists a process to utilize this data, to really tell people whether they have genomic risks. And we have chosen a pilot project where we have 64,000 patients' data, and it's from the FinGen Data Freeze 4, which was a couple of years ago. And we have chosen these mutations, so the easy ones, I would say. So BRCA1, BRCA2, and PALB2, which is highly enriched in Finland. And why? It's because there is a consensus that when having this genomic mutation, you truly have a risk. In this case for breast cancer, 40 to 80% in case of BRCA, and 30 to 60% risk for PALB2. So there will be not too much objections to why we are doing this. And also the other reason, of course, being that these are actionable variants. So we are actually piloting the whole process chain. So we are identifying, validating, recontacting, and the hospital is counseling the patients. And we are only transferring the true positives to the healthcare. We are also collecting feedback from the patients during the process. And based on the results, I think this will be implemented nationwide. Remember that in FinGen we have 500,000 people, so 10% of the population. So it's potentially a big number of patients. And the results will be published next year. So I'm not able to tell you all the results, but I can tell you the process. The process starts that in that we look from our database and look for the genomic data relevant for this particular case. We identify the sample donors because we of course know their true identity, and we screen out those people who are already known to be carriers in the healthcare system. Then we do internal validation because the axiom genotyping array is not really a clinically grade acceptable way of genotyping people. We are actually using a clinical grade analysis method using the hospital genetics department. Then we recontact the patient, and what we ask is actually the permission to transfer this finding, or can we transfer the finding to the healthcare? So this step is only to ask whether we can do that. And if they say yes, and to our knowledge, so far about 2/3 of the patients contacted say yes. And if you look at the numbers, I mean, you can check from literature that it's about 1% of the patients in this particular cohort that will really have this variant. Then we transfer the patients to healthcare, or they are not necessarily patients. And actually what we have found that 80% of the people who we're contacting don't know, and the healthcare doesn't know that they are carrier of these mutations. And what then happens is that the patient is recalled to give another sample to make sure that the biobank has not screwed up and has somehow mixed the tubes. And well, we haven't so far, which is very good. So we have only delivered the data from true positives. And then what happens next is genetic counseling, and then follow-up and potential care. So this is ongoing, as I told you. We are also collecting feedback, and some feedback is shown here. So it is surprisingly positive. So we are afraid that, okay, some people might not like it. We haven't had many people who have kind of signed out from the biobank consent. So this is very encouraging, of course, for us to continue. And it's very important to notice that none of the responding individuals has regretted that they participated in the biobank project. So that's the situation now, and hopefully next year then you will hear the complete story with all the numbers and images. Thank you very much.
Okay. Thank you. A very interesting story, actually. Let me ask you the first question, because we went through the similar procedure using hereditary hemochromatosis as a mutation for hereditary chromatosis as a point, and of course that was also interesting. But my question is, okay, let's say what do you see are the next steps? So because at one point the most difficult decision would be the list of the potential mutations or diseases that kind of would be approved for the reporting.
Yeah.
Have you thought about that?
Of course,
is it feasible or?
Well, of course, first we have to prove that this is feasible. And so far we haven't heard so many objections to why are you doing this and this is useless. But of course the difficulty comes if we are talking about risk factors of other genes. And I don't know, we don't have a plan for the follow-up really. And actually we have to of course listen to the doctors and also the patients and be really careful in returning the data otherwise. I know that our friends in Estonia, for example, they have done this type of things already for a number of years. But it also takes a lot of capacity from the healthcare. And actually one study we are doing at the same time is estimating the cost to healthcare. Because if you say it's 1% from 500,000 people, that's 5,000 people to be counseled. And so it's also a cost burden to the healthcare.
Yeah, I think it's a challenge. So other questions?
I have a question. I think that's really fascinating what you're doing here. So when you're returning back the data, is it then possible then for these patients to submit these data to other analysis pipelines so that they submit their sequencing data and run it through different pipelines which are available?
And then they would give it to Biobank. Well, it hasn't happened so far. And I don't think we have a process for that, for taking in people-generated data. I think that would be useful for a certain project. So if a project wants to use our samples and data, then why not? Why not use also patient-reported data?
And the next question, I think you need a high level of education in a way to tell the people what does this data mean? What does it mean that you have a risk gene or something? Do you have any educational program, or you're just doing it like that you're telling the people when they're coming to you? And,
well, we don't have the resources to give any education to the people, ordinary people. But of course they will learn. Those who are contacted, of course they will learn. And based on the feedback, the learning procedure is not so unpleasant.
Impressive. Yeah.
Thank you.
Okay. Actually, one final question. Do you have any feedback from the... actually, can you access those people who didn't agree or didn't respond to this kind of share or transfer of the data to the healthcare sector?
Yes, so we are now making a second round of... We first sent the letters, now we're actually trying SMS messages.
Okay.
Because some people don't read the letters, or maybe the post office doesn't work.
Because I think that this is very important to get at least the feedback why they refuse this, or whether it's just not being informed, or is there some other reason.
Yeah, the first round was maybe 60–70% responses. Yeah.
Okay, thank you. Okay, our next speaker is Peter Paediatric from the University of Tartu. He is Associate Professor of Oncology, also a medical oncologist and healthcare entrepreneur, and looking at the cancer from different angle, from the taking into account the polygenic risk scores. Yeah.
Thank you. Dear colleagues, ladies and gentlemen, my presentation goes beyond from biobanking, and my topic is actually precision prevention of common cancers, focused on use of polygenic risk score technology. Here are my disclosures, and we all know that cancer is a major healthcare problem. And actually, our risks for everybody to get cancer during our lifetime is actually quite high. We can win many cancers using prevention and early detection, and for that, cancer screening programs are implemented. But currently, these programs consider only age of people. Like in case of breast cancer, mammography screening from age 50 to 69 mostly, but actually our risks are very different. And new solutions could be personalized risk-based prevention and screening. And we need tools for that. And genetic background actually is very important component in case of many common cancers. You know that already in use in healthcare are tests to detect so-called single gene mutations or monogenic pathogenic variants. Like here in case of breast cancer, this yellow cloud of genes. but they are relatively rare. And research shows that many single nucleotide polymorphisms also influence our cancer risks and also risks for other diseases. And this part is not yet in use in healthcare. Impact of every SNP is relatively small, but they are common. And it's possible to analyze the complex impact of SNPs using polygenic risk score technology. Our approaches are based a lot of Estonian biobank activities. We have in Estonia 200,000 individuals included with genotype data and also collected phenotype data in biobank. And 2018 to 21, we had a national clinical pilot project where exactly was done feedback to biobank participants regarding breast cancer and cardiovascular disease risks. And we used in this approach not only monogenic variant feedback, but also we started to use polygenic risk score estimations. And 890 women with high polygenic risk score were invited to this project, and also 109 women with detected moderate or high risk monogenic variants. And actually, the study modeling showed that with such approach, we can save 14 to 22 lives in Estonia every year. And with quite cost-efficient way, so the quality-adjusted life year cost is a little bit less than 7,000 euros, which is actually quite cost-efficient. And also feedback from participants shows that this information was received quite well from participants, and we saw that most important thing is to give exact recommendations for next steps, what to do with high risk levels. Estonian data, for example, in case of breast cancer shows that we have approximately 2% among women of carriers of monogenic pathogenic variants, but we have up to 20% of women who have moderate to high polygenic risk level. And we know that actually such personalized screening is already implemented with international and national guidelines for monogenic mutation carriers. But actually it's quite clear that for all those high-risk women, we should implement screening differently than is current one-size-fits-all approach. From May this year, in European Union is in full force so-called in vitro medical device regulation, which says that genetic tests in European Union should be in the future so-called CE-marked in vitro medical devices, or lab-developed tests which have quite strong restrictions also from regulatory side of view. And this was exactly the reason why we founded a company, Antagenes, which is a spin-off company from University of Tartu, with aim to develop and bring polygenic risk score technology into use on clinical grade level in healthcare according to healthcare regulations. And currently, we have developed and brought into use as healthcare services 4 tests for breast, colorectal, prostate cancers and skin melanoma. And in case of breast cancer, for example, you can see this green line here is average breast cancer risk in Estonian population, but we can separate high risk level and also low risk levels for breast cancer. Of course, the important thing is not genetic testing or risk estimation, but actually the aim is to reduce cancer mortality using personalized prevention programs. And for example, in case of breast cancer, all women are invited currently for mammography screening from age 50. And mammography after every 2 or 3 years in most countries, and further management in case of mammography findings. But looking for Estonian data, we see that this screening group covers only 1 proportion of breast cancer cases. Younger women with higher risk levels are out from this screening. And at the same time, it's not reasonable to screen all younger women. And actually, the new model for that, therefore, which we are already implementing, is that women from age 35 are offered genetic testing both for polygenic risk score and monogenic pathogenic variants. And according to these risk levels, we can give recommendations when to start screening, how to do that, and what other possible methods or steps are possible to use to reduce risks. And in implementing polygenic risk scores, there are such principles like equivalence of risk. So principally, societies have agreed that average risk level at age 50 is good enough to start mammography screening. But if younger women have already higher risk level, then actually they should also receive screening earlier. And the other principle is equitability. So if some persons, they have access to prevention and screening, then actually other persons with similar risk levels should have also similar access. And maybe third principles regarding polygenic risk score is similarity of moderate risk monogenic mutation carriers like CHEK2 and ATM, for example, because polygenic risk score shows similar risk levels. And we know that there are already international and national guidelines for these mutations, and we can implement similar approach for polygenic risk score. And here, for example, we created such first guidelines for polygenic risk score recommendations, and we do not recommend to reduce current screening, because we think that to recommend that, we need randomized studies to say that this is safe. But we recommend start screening when risk level reaches average risk level at age 50, and we recommend more intensive screening with those women who have higher risk levels. From this year, Estonian Health Insurance Fund, who is such payer in Estonian healthcare, accepts our polygenic risk score recommendations for breast cancer screening. So if we give these tests into healthcare, then women can use mammographies which are already covered by health insurance. And we have also implemented such model for breast cancer precision prevention. We have ongoing international projects also in Norway, Sweden, and Portugal, where we testing similarly using such implementation research methods, similar approach in these countries. And principally, I can conclude that here is principally the value proposition from polygenic risk scores for healthcare, that if we can detect more precisely risks, then we can implement precision prevention programs and reduce with that cancer mortality. So principally we can say that the future of such personalized precision prevention is already available just now, and of course there are a lot of questions to address still, and we are looking also for international research and implementations, cooperations in this field. Thank you very much, and be healthy.
Okay, thank you. Any question? Okay, I definitely have a question, because it always puzzles me how to deal with the communication with the patients, especially in this area, how to report these polygenic risk scores if we think about the absolute value of this risk score. Because imagine that in 2 years when we have more association studies data, so you recalculate this risk score and it will definitely be different, and maybe for some people you fall out of this increased risk group. I wonder if you have thought about it, how to really communicate this to the people, and how often you actually should like do this recalculation or yeah.
Yeah, it's a very important question, absolutely. We have dealt a lot with this, actually. And we have created such quite detailed and long report to explain these aspects. And this is, Oviedo Convention says that in case of every genetic test, the patient should have access to counseling. But it's not, we are in the process of such broader implementation of genetics in healthcare. And in every case, maybe it's not necessary such doctor consultation, but it's possible to use also in written form or videos or different forms of counselings. But exactly this information should be understandable. And therefore, we have such long written report with pictures explaining the meaning of these test results. And we can give a comparison, as polygenic risk score is actually comparison with other people in same population, in same age. Then we give this comparison and try to explain visually what are the risk levels. This is absolutely very important question. And also for those individuals who want additional counseling, we offer this possibility also. We use currently telemedicine solutions for that, so no need to come to doctor's consultation, but it's such video consultations, they work quite well.
Okay, thank you. Other questions? Okay, if not, but really congratulations to Estonia that you were managed to bring it to the next level, really, and implementing this in the screening. Of course, I think it's very rarely that it is done now on the polygenic risk score base, and I think it's very important to perform such studies to really see the efficacy of this approach.
Actually, we have currently a similar problem that we have 200,000 individuals with genotype data, like a similar problem like in Finland. And we had a lot of questions from our patients that why we have to do again genotyping, because we use exactly same method as is used in Estonian Biobank. And currently, there is project in Estonia which is working out the ways how it's possible to transfer biobank data to healthcare biobank. Individuals, they have to give additional consent for that. And then, principally, for example, such test providers as we are, we can use already this available biobank data and give test results according to healthcare regulations based on earlier genotyping. Of course, this earlier genotyping must be done according to quality criteria, what are necessary for healthcare use. This is why we have this in vitro medical device regulation in Europe. But we actually thought what to do, and then we implemented such, how to say, finally, not a very patient-friendly solution, but this was the best we could do. And this was so that if biobank participants, they asked their genotype data out from biobank, then biobank gave this to them we have such possibility to crypt data and decrypt with identity card in Estonia. And so they received this crypted genetic data, they decrypted themselves this and uploaded to our server. And so we could then give also these test results to using this already available biobank data. And actually we received such Sitra Innovation Fund in Finland made competition of fairly data use in healthcare. We were one of winners 2021, I think, about this approach. So I think, but similar questions we see in every country, and this should be solved so the regulations are followed, and also a clinical impact is there. Thank you.
Okay, thank you. Okay, we can now move to the next speaker. Next speaker is Ilmārs Stonāns, who is Deputy Director at Institute of Clinical and Preventive Medicine, University of Latvia. Yeah, floor is yours.
Thank you for your introduction. I'm here with another story, another example of biobanking. In this case, it's a story about how biobanking emerged from cooperation between hospital and academic partner. In this case, Institute of Clinical and Preventive Medicine, University of Latvia, and the Riga East University Hospital. Next slide. Okay. I'll begin with this slide, actually, the ideal biobanking from the perspective of the patient pathway. It shows how many possibilities of sampling do we have for biobank on the patient pathway through cancer therapy. So prior to therapy, directly before surgery, during the follow-up therapy, and the possibilities to use these samples not only for biobanking itself, keeping in biobank, but also the possibilities to use these samples for further analyses in disease models or as derivatives for sequencing and so on. It's an ideal situation, and this would be perfect to have something like that, but it never happens. That's why me myself prefer to keep things simple And I have simplified here the process of biobanking, with the biobank being somewhere in the middle of this process. Because initially patients or healthy people are there who agree to donate their blood tissues, biofluids, or microbiome too. And then these samples are stored in the biobank, which itself is a framework dealing with quality management, ethical, legal, compliance, process management, with a lot of SOPs in place. And then two main components of biobank are there. On the one hand, samples which have to be collected, stored, processed, and at the end of the story distributed. And another part, sometimes even more important part, these are metadata about these samples, all the possible additional data about medical history, imaging, histology, lifestyle, and so on. It has today already been mentioned that biobank is somewhere in the middle between hospital and research. In our case, I'm presenting you an example where hospital and research came together, and it resulted in the collaboration for biobanking, specifically for biobanking for cancer patient tissues. Historically, it began about a decade before, and as usual, finances are important, and financial support came from European Social Fund project, interdisciplinary research group for early tumor diagnosis and prevention. This resulted later on in formal establishment of tissue center within Riga East University Hospital. And a couple of years later, when our Institute of Clinical and Preventive Medicine of University of Latvia was established, we already had biobank as one of our structural units. This model brings the best possible competencies from both, from hospital and from academia, and it has to be mentioned that hospital surely ensures proximity to patient, clinical competencies, and the possibilities of pathological lab, along the other good things you can have in the hospital. On the side of academia, of course, the research competencies are stronger, as well as experience in additional acquisition of financing, which is always important to have a biobank. And in our case, also, data system management is provided on the side of university. During these years, altogether collection of samples, altogether biobank participants are a bit more than 8,000, and 4,300 of them are cancer patients. Of course, we do have all the necessary hardware in place, partially on the side of hospital, partially on the side of our institute, and these are altogether more than 20 freezers. We have the possibility for liquid nitrogen storage, all the necessary small hardware for sample coding, and sample processing before biobanking, as well as equipment needed for DNA isolation, and also possibility of sampling of breath samples. We have chosen for information system the solution based on a Microsoft SQL Server, which is located in hospital, so all the data are physically present there and preserved and isolated from others by firewall. The users of the information system can use it through Microsoft Access-based information system. Microsoft Access was chosen because of its flexibility to generate different types of data entry models. What we have learned from our experience is that if we want to collect the largest possible information about the patients, beginning from questionnaires and including all the possible medical data, then we can't have a unique data entry model for all kinds of cancers, for all types of cancers. That's why they differ a bit depending on the initially diagnosed type of cancer. Here are two collections of biobanks, or two biobank collections, which are running under the administration of our institute. On the one hand, the first one is Clinical Biobank. We are running in cooperation with Riga East University Hospital. As already told, it has 8,300 patients already in it, with more than 90,000 different samples collected. And in parallel, we also have in the institute another population biobank project, biobank based on gastric cancer screening. It's even larger, with 10,000 participants and more than 200,000 different samples. We try to collect as much different samples, types of samples as possible. As usual, the standard is blood, whole blood, sera, but also we are collected quite regularly fecal samples, and for the cancer patients undergoing surgery, those are also surgical samples, frozen surgical samples, or biopsy samples, as well as saliva and exhaled air samples, because our institute is dealing with volatile markers of cancer. And if getting back to our clinical collection of cancer patients, then we see that it is the largest one if we are dealing with a biobank run together with Riga East University Hospital. The largest proportions of all clinical cases are cancer patients, with the largest collections being for stomach cancer, colorectal cancer, and breast cancer.
Of course, this would be useless
to have a biobank without using the samples. And these are the examples of the projects linked to biobank. I'm using the term linked because these projects are not only using the samples we have collected. The majority of these projects are also contributing for the collection of the new samples for biobank. A lot of them being, as already mentioned, linked to our interests in volatile markers. We also, in the last time, engaging us in Million Microbiome of Human project. Ethical aspects of biobanking have been also attributed, as well as lung cancer markers. We are proud to be part of Latvian Biobank network, along with other colleagues being present here today too. And we are also proud to be listed in BBMRI ERIC directory along with another Latvian biobank genome database of Latvian population. And thank you.
Thank you for this really informative presentation. Any questions?
You see, I have a question. I was impressed about your breathing samples. So, I mean, there must be a large volume. So, how you store them at -80 freezers, or
sorry?
The breathing samples? So, how many samples do you have from this breathing?
Okay, there are different methodologies to collect them. There are solvents which are used to collect them, then they can be desorbed and used for analysis, for example, with gas chromatography, mass spectrometry. Another possibility is simply to collect the samples in the specific bags. Most of these samples are already used within the research project, so they can't be really stored for long times.
Okay, we have a question from the audience.
Does it work? Yeah, now it works. I saw today in a row many presentations informing about how many biobanks in Latvia are there already. Just as a concept, is it imaginable to merge those into one biobank? Would it be maybe cost effective, or is it unpractical? What maybe are there any points for discussion? We have some guests here from other countries, so maybe you have a comment, Dr. Baumbusch, about this. Do you think every clinic should have its own biobank? Is that good?
What this question to me?
To everyone for discussion. I just thought.
Okay, of course I have an opinion about that. And we had also with Jānis a couple of years earlier discussion about that. Of course, centralized model has good sides for it. On the other hand, centralized model can't always take into account all the clinical aspects. And as I mentioned, I believe that simply to have sample is not enough. What we are facing from day to day is, do you have samples? Yes, we do. But can you tell us if these samples are coming from cancer patients? Yes. Are they coming from gastrointestinal cancer patients? Yes. Are they coming from colon cancer patients? And we can go deeper and deeper. Are they from women? Are they from children? Are they from, how old are the patients? Did they have chemotherapy, forehead or not? Genetic family history, so on and so on. So these metadata are extremely important. And you can't have, for all types of clinical cases, unified metadata templates. So I believe the best solution is the cooperation between centralized biobanks where it's possible. And it's definitely possible for the genomic databases. In the fact, we are cooperating with a genomic database of Latvian population, and we are sending a lot of our samples to Biomedical Center for them to perform all the possible sequencing procedures so we can complement each other. On the one hand, clinical data and tissue samples. On the other hand, genomic data.
Yeah. If I can comment on that, I think also that first we should be very practical, and I think really see what is the most cost-efficient variant. Not only just thinking of the really putting everything in one place. If it's not effective, it also won't be really feasible to do so. And I think that which is really most important question is not actually where the samples are kept, but to follow some principles for the quality control, which is possible to establish in this network. And this is one of the goals of the BBMRI, actually, to really to at least to understand what are these quality issues to deal with. And of course, the next level is the data. If you can exchange freely with the data and that they are harmonized between biobanks, so that already is a major goal to really be. And I think we are going to the right direction.
Thank you.
Okay, I guess this is all for today. First of all, I would like to thank all the speakers, and we are almost in time, only 5 minutes late, so this is amazing, I guess. Thank you, and also for all the listeners that are here, and see you tomorrow. Okay, thank you.
Thank you.